Hello, everyone. Welcome to today's live broadcast, The Hidden Effects of Epigenetic Discoveries. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Rockland. On the call is Dr. Karen Abarca-Heidemann, Rockland's Director of Research and Development.
Hello, everybody. Rapid advances in the field of epigenetics are revealing a molecular basis of how heritable information other than DNA sequence can influence gene function. Epigenetics findings add to our understanding of transcriptional regulation, nuclear organization, development, and disease. Rockland, as a manufacturer of research tools, understands the importance of these scientific advances and is proud to support and work together with scientists like Dr. Kim, Dr. Barrero, and Dr. Scharer that will be presenting today. Rockland supports academic, biopharma, and diagnostic industries with antibodies and antibody-based tools for over 50 years.
Thank you, Karen. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's speakers, Dr. Jeffrey Kim, Dr. Carlos Barrero, and Dr. Christopher Scharer. Dr. Kim has worked in oral health research for the last 10 years and has a broad perspective of the field. He received specific training in stem cell biology, regenerative medicine, neuronal specific differentiation, bioinformatics, next-generation sequencing technologies, salivary gland biology, developmental biology, chemical toxicology, drug abuse, and addiction. Dr. Kim has extensive experience in analyzing embryonic cells and tissues in the context of identifying the regenerative potential and profiling the genome and epigenome. Based on previous collaborative projects, he has developed great working relationships and strong communication skills with his collaborators, and has established strong ties with experts from oral biology and bioinformatics in government and academic research institutions. His current projects address repair and regenerative mechanisms in the context of oral health. Dr. Carlos A. Barrero is a research assistant professor at the School of Pharmacy at Temple University. His research interests focus on applying proteomics, metabolomics, and bioinformatics technologies for the study of pathological mechanisms of different diseases. Dr. Barrero has made substantial impacts to the knowledge of pathological mechanisms underlying COPD. His current interest is to explore the ability of H3.3 to be used as an early marker and as a therapeutic target for COPD progression, which will have a direct translational application and will assist greatly in the identification and treatment of patients with this disease. Dr. Barrero has made similar contributions in the areas of obesity and in HIV/AIDS. Christopher Scharer received his BSc and PhD from Emory University in Atlanta, Georgia. During his graduate work, he studied the role of oncogenic transcription factors under the mentorship of Dr. Carlos Moreno. In 2009, he joined Dr. Jeremy Boss' laboratory as a postdoctoral fellow, later progressing to instructor in 2014, and more recently, assistant professor. In Dr. Boss's lab, his research focused on identifying epigenetic mechanisms of T and B cell differentiation. Currently, Dr. Scharer's research focuses on improving and adapting sequencing applications for low cell input numbers, including ATAC-seq, ChIP-seq, methyl-seq, and RNA-seq. He currently leads an effort to integrate these epigenome mapping technologies to understand normal immune function and autoimmune diseases. Through identifying pathogenic epigenetic signatures using a systems biology approach, he aims to reverse engineer the environmental stimuli that impact human disease, and discover novel biomarkers and therapeutic targets. I will now turn it over to Dr. Barrero for his presentation.
Hello, everybody, and thank you, Judy, for your presentation. I also want to thank Rockland for the invitation to present my work in this webinar. Today, I'm going to show you an unconventional function of the histones that has been recently studied in the past few years. But first, let me show you a brief introduction of what histones normally do. They are, as many of you know, as maybe all of you know, histones. There are several class of histones. There are 5 families: H2A, H2B, H3, and H4. All of them build up a nucleosome that is the basic structure of the chromatin to wrap around and condensate the DNA in the nucleus. These histones have amine and C-terminal tails that are highly modified in different amino acid residues, and the modifications include acetylation, methylation, phosphorylation, citrullylation, ADP ribosylation, simylation, and ubiquitination. The main function of this histone is to keep the DNA wrapped around and as seen here in the presentations, and allow the gene expression when they are required. Then, by modifying the histone tails, there are several enzymes that are key regulators of this process in a context of epigenetics. But this is when the histones are present normally inside of the nucleus, inside of the cells. What happens when the histones are present extracellularly outside of the cells? Let me tell you, the histones can be cytotoxic for the cells, and they can produce injury in different tissues all around the body. They can induce inflammation through specific receptors, including toll-like receptors, and these extracellular histones are mainly released by immune cells that are activated, like neutrophils and macrophage, in different process of activation. For neutrophil, it's called netosis, or they can also be released by cell death during apoptosis or necrosis. These cells, the extracellular histones, also induce itself apoptosis, as we described in our work, through in a receptor that is still unknown so far. Including the lungs, also there are several tissues that get affected by histones, as in the brain by stroke, liver injury, and the kidney injury. The histones can induce also pancreatitis, peritonitis, and also has been described to be implicated in the retinal detachment. For the purpose of our conference, we are going to go deep in the study of how histones or extracellular histones are related to the pathogenesis of COPD. A little bit about COPD. COPD is a disease that is characterized as an inflammatory chronic disease that has intensive destruction of the lung tissue, mainly in response to smoke. And what has been known for decades is that the cigarette smoke induces increase in the intracellular reactive oxygen species, and these reactive oxygen species modify the histone deacetylate enzymes, allowing the histones to keep acetylated and inducing active transcription of the inflammatory genes. Our recent work add to this knowledge additional arm of this mechanism, in which the inhibition of the HDAC contribute to the accumulation of the hyperacetylated histone 3 that is resistant to be degraded, and when the cells die, they get released to the extracellular space, inducing cell toxicity and being deleterious for the surrounded cells, promoting the COPD progression. Let me show you how we did this in detail. Initially, we performed a proteomics study using label-free analysis in which we compare the COPD lung tissue against the control tissue that become from normal patients. And when we perform this proteomics analysis, the protein that highlight with the significant difference was histone 3.3 with a-fold difference. Then we decided to characterize these proteins and study deeply. First, we did a confirmation of the histone 3 in the tissues that were subject to the proteomics analysis. And we did a validation study in a large number of samples of different degrees of COPD. Very severe, known as gold 4, or moderate COPD, gold as 9.3. And they were very significantly increased levels when they are compared with the control group. Then we decided to look where the histones were located inside of the tissue, and for our surprise, we were able to find the histones that were extracellularly in the air space, and more importantly, they were interacting with the bronchoepithelial cells. We also were able to identify these free histones in the bronchoalveolar lavage and in the plasma, giving an indication that these histones can be a marker for COPD. Then we decided to evaluate what was the origin of the overexpression of the histones. Initially, we test the transcription of the different genes of histone 3.3, and we didn't saw significant difference. But when we evaluate the degradation of the histone 3.3, we were able to find isoform of the histones that are ubiquitinated. And applying the proteomics technology, we identified the specific residues where the histones were modified by ubiquitination. Then we also, applying the same technology, were able to map the whole modifications in the protein, in the histone 3, and we found that the histone 3 is hyperacetylated in the end term of the histone 3. And this finding was confirmed by blood in independent subjects of COPD, and all the subjects with COPD have hyperacetylated histones compared to the control subjects in the control groups. More importantly, we are able now to differentiate patients that have COPD by measuring the histone 3 and its modifications, the acetylation of the histone 3. But the importance of this histone 3 to be extracellularly in the lung tissue was then assessed by different experiments leading to demonstrate that the histones were apoptotic to primary bronchoepithelial cells. And we were able to, initially we measured the incorporation of annexin 5, and we saw that the histone 3 extracted from the lung of the patients were cytotoxic. Even the normal histones that is not modified, doesn't have any acetylations or modifications whatsoever, is still very cytotoxic. This histone, the H3.3, is more cytotoxic than the rest of the histones. And this cytotoxicity is dose dependent. Okay, one of the clear experiments that we did to demonstrate that they are cytotoxic, we overexpress the histones in intracellular weight or in a form that is secreted. When we saw the histone that is secreted into the media, we saw that the toxicity was about 70%. Only 30% of the cells survive. And more importantly, this cytotoxicity can be prevented by the specific antibodies that recognize the N or the C term of the histone. Then this later we confirmed by Western blot that the apoptosis induced by histones in these primary cells are through activation of caspase, and we perform a proteomics analysis on these cells. And to summarize the finding of the mechanistic that are implicated in the cell toxicity due to H3.3, we found activation of the calcium signaling pathway, and we found activation of the unfolded protein response pathway, that is a stress response that the cells produce. And we also found mitochondrial cytotoxicity. We performed several flux studies, and we were able to see that immediately after we put the histones in these primary cells, there is a flux of calcium that activate the release of calcium from the endoplasmic reticulum. They get incorporated into the mitochondria, and this event happens several times after the histones are in contact with the cells, really finally doing damage on the membrane potential of the mitochondria and releasing different caspase, or activating apoptotics also through an independent way downstream of UPR. Finally, when these cells are dead, they release more of the acetylated histones that were accumulated that are resistant, and they perpetuate the cycle of death. Then the implications or the application of our work have now linked the histone 3 as a possible cause of the inflammatory response that characterize COPD. We are now doing another experiments using proteomics technology to quantify histone 3 in plasma and use histone 3 and its modification as a biomarker for COPD. And we are also developing some assays trying to look for molecules that can be used as a pharmacological approach to prevent the histone 3 toxicity. But let me finish my presentation showing an unpublished results that we have done to demonstrate the in vivo toxicity of histone 3 and the toxicity of the histone 3 acetylated. Basically, we instill the recombinant histone 3 or the acetylated isoform of the histone 3 into the lung of the mice, and 48 hours later, we evaluate the lung tissue damage by different techniques. Our results, initially, we just confirmed that the histones are present extracellularly in the parenchyma of the lungs, as well as interacting with the bronchoepithelial cells just 1 hour later after we do the instillation. And 48 hours later, we see dramatic change in the epithelium of the controls compared to the histone 3. You can see enlargement of the bronchoepithelial cells, as well as infiltration, cellular infiltration in the parenchyma of the cells with destruction of the walls of the alveoli. These results are more severe when acetylated histone was instillated into the mice, and these results were also confirmed that they are inducing apoptosis, as shown in this immunohistochemistry by caspase 3 and caspase 9 activated, and the cells that are dying are basically the cells that make the bridge in a normal alveoli function. Then we confirm our results, the anatomical findings in using micro-CT, where you can see in this reconstruction of the lungs of the mice, the more severe damage of the lung is caused by acetylated histone or hyperacetylated histone that we use. And more importantly, we were able to characterize the inflammatory response where we saw that different cytokines like interleukin-6, TNF-alpha, MCP3 are highly expressed in acute form after the long inoculation of the histones. For example, what happened into the COPD patients. Okay, now to conclude, I want to summarize what I have presented today. There are different sources of histones that can be from immune cells activated or from dying cells through apoptosis or necrosis. They are initially leaded to kill bacteria, but they also can be damaged for normal tissue, and the consequence they can induce a lot of injury and inflammation in this specific tissue. They can also be induce activation of platelets and induce thrombosis. They can also predisposed to autoimmune disease by the release of the DNA that is attached to the nucleosome. But the good news is that there are several molecules that are known already that are able to degrade or to block the histone toxicity, and we are looking for more. Okay, thank you so much for your attention. I want to acknowledge all the collaborators here at Temple University and Temple Hospital, and this work has been supported by NIH grant and Flight Attendant Medical Research Institute. Thank you.
Thank you, Dr. Barrero, for that informative presentation. I will now turn it over to Dr. Kim.
Good afternoon, everyone. My name is Jeffrey Kim. I'm a project leader for ADA Foundation in Gaithersburg, Maryland. I would also like to thank Rockland for giving me this opportunity to speak to you today. These are my learning objectives for today. How could epigenetic analysis enhance your research? What high-throughput epigenetic analysis are currently available? And tips on sample preparation and choosing a core facility, and what to do with your raw data after they come back from the core facility, and also correlating epigenetic data to gene expression and downstream application as well. And I'll be using our recent publication to give specific examples on how these learning objectives can be applied to your own research. So the areas of research that may benefit from epigenetic analysis are cancer, aging, developmental biology, stem cell biology, toxicology, pharmacology, and environmental toxicology, brain function and mental disorders, and many, many, many more. The currently available high-throughput epigenetic analysis include DNA methylation, including MDIP sequencing, restriction-based methods, bisulfite sequencing, as well as histone modification analysis, including chip sequencing using histone marks, RNA associating silencing analysis, non-coding RNAs, PU-associated short RNAs, and microRNAs. I'd like to point out that DNA methylation and histone modifications, the ability of specific antibody that chip grade is very crucial for success of this analysis. So tips on sample preparation, genomic DNA for MDIP sequencing or chip sequencing, you have to have a clean DNA prep without RNA, DNA, protein contamination to begin with. And you like to check that, do a QC control before sending out to the lab. Fragmentation step is also important, and you want to do it so that there's no heat damage on ice and short sequence. Range of DNA size should be checked before sending out to the lab as well. The IP step has to be performed with a chip-grade antibody, and the quality control with chip qPCR before investing costly sequencing would be advised. And again, IP step may require some optimization in terms of buffer choices, incubation time, and number of washing steps, and so on. Tips on choosing a core facility. Obviously, a local core facility is preferred. They may help you in sample preparation and running some QP analysis, as well as post-sequencing data processing. Experienced core facility with publication record is highly recommended, and sometimes it's difficult to find a core facility near you. In that case, it's also possible to look for a good one from a high-impact publication. They're usually mentioned in the acknowledgement section. So what to do with your raw data once it comes back from the core facility? So you want to establish a post-sequencing plan with a bioinformatic expert before sending out your sample, and I think that's one of the common mistakes that people are sending out the sample not knowing that how to analyze the raw data once they receive it. And second thing is raw data, usually they're in a FASTQ type of file, and they're not aligned to the reference genome yet. So you need to use short read aligners such as Bowtie to align your raw data to the genome. And once the file is aligned to a genome, usually they're in SAM type of format, and they're ready to be used for further analysis. So what to do with the raw data, part two? First thing is you want to perform quality control experiments again using a line file. And you want to subtract the background noise using IgG control, and you want to identify enriched region. It's also known as peak calling, and there are numerous applications and programs that you could use, and you annotate the enriched region to the nearest genes. So from this point on, I'm going to use specific examples to further understand our learning objectives for today. So before considering costly sequencing analysis, the ELIJA type of assays, they're very inexpensive and easy to do, and you can get a lot of good information from that before doing the sequencing analysis. So for example, here we did alcohol treatment on human embryonic stem cells in a different concentration, and then we looked at 5-methylcytosine percentage using ELIJA. And then we realized that alcohol does change DNA methylation, the global DNA methylation. So at this point, we were ready to do further analysis and targeted approaches. So it's very important to correlate your methylation data to gene expression. So what you're looking at here is the y-axis represents fold change using a microarray, and the x-axis represents DNA methylation values. And the green dots represents the genes that we're interested in. So their fold change with alcohol treatment increased, and their methylation value decreased. And the yellow dots represents decrease in gene expression with alcohol treatment and increase in methylation. And another way to represent the same data is the left blue box represents gene expression and methylation change upon differentiation of stem cells, just normal differentiation. And the right green box represents gene expression and methylation change upon alcohol treatment. And then the middle is subset of genes that we were interested in, that alcohol-induced differentiation related to alcohol-induced differentiation related genes. And next, performing a functional validation is very important. So the blue bars represent control cell expression, and the red bar represents alcohol decreasing that gene expression. And the green bar represents 5-azacitidine treatment cell expression. And as you may know that 5-aza treatment inhibits DNA methylation. So here, by controlling the DNA methylation, we were able to revert gene expression change due to alcohol. So having a functional validation is very important. There are many different types of downstream applications, and one example is finding relevant transcription factors and common motifs. And we use a STEM program to further understand alcohol's epigenetic effect on stem cell differentiation here. So hopefully my presentation achieve our learning objectives today, how we could use epigenetic analysis to enhance your research, as well as some of the high-throughput epigenetic analysis that are available today, and tips on sample preparation as well as choosing a core facility, and when the raw data comes back from the core facility, what to do with them, as well as correlating epigenetic data to gene expression and some of the downstream applications. So thank you for your attention. That's my email. And if you have any questions, feel free to email me. And our project was funded by NIAAA and CERM from California and NIDCR training grants. Thank you very much.
Thank you, Dr. Kim, for that great presentation. Next, we'll hear from Dr. Scharer. I will now turn it over to him.
Thank you. Hi, my name is Christopher Scharer, and I'm at Emory University. And today I'd like to thank Rockland for giving me the opportunity to present my research. And I'll talk to you about the role of epigenetics in regulating the timing of gene expression during B-cell differentiation, and some of our work using some emerging application or mapping technologies for epigenetics. So our lab works on the ability of your body to make antibodies. So B cells, which are quiescent cell type with antibody bound to the surface, are able to differentiate into plasma cells upon recognition of antigens. Plasma cells are long-lived cells that are dedicated to antibody secretion and can secrete up to 10 to the 7 antibodies per second. Antibodies can last for up to the lifetime of the host and are good in that they can protect you from infection from influenza, polio, but are bad in circumstances where the production of antibodies to self-antigens, such as in autoimmune diseases, is pathogenic and problematic. And so understanding how this process of differentiation from B cells to plasma cells is regulated is important. So we're interested in what the epigenetic processes are that regulate this differentiation process as the cells go from a quiescent cell type to a cell type dedicated to making antibodies. And with the goal of, can this process be manipulated for both good in times of when we want to induce these plasma cells, and in times when autoimmune diseases, can we develop targets to inhibit pathogenic plasma cells? So we developed a model of B-cell differentiation in the mouse in vivo. We did this by taking B cells, purifying them from a mouse, labeling them with a division dye called CFSE that allows us to track which cell division they are in. And also these B cells contain a congeneric marker called CD45-1 that allows us to track the adoptively transferred B cells. We put these cells into a mouse, a mu-MT mouse, which does not have B cells, allowed them to home for 1 day, and then we inoculate these mice with a bacterial cell wall component, LPS, and look at the phenotype and the response 3 days later. On the bottom, you can see flow cytometry plots of congenically marked B cells. So the CD45-1 marker on the x-axis shows that we can find the B cells that we transferred. On the right plot, you can see what happens in a mouse that was not injected with LPS, and that all of the B cells are positive for the B220 marker on the y-axis, which is a B cell marker. However, when we differentiate and look 3 days later after LPS injection, we see the formation of plasma cells as shown by acquisition of the CD138 cell surface marker, and that the plasma blasts in the red box are cycling cells, whereas the plasma cells in the orange box are post-mitotic cells. Now, when we look at division using the CFSE dye in these cells, one of the things we notice is that these cells undergo many rounds of cell division before they acquire differentiation markers. So we can pick out the division states of each cell such that the farthest to the right is cells that have the dye, and that every division the dye is diluted twofold. And so we can physically track cells that have gone through 1, 2, 4, 6, or cells here which have gone through 8 or 9 divisions before they acquire the CD138 marker of plasma cells. And this was intriguing to us, as we did not know in vivo that these cells went through this many divisions and that there was a lot of division required before differentiation. In order to understand what was going on at the molecular level of these cells, we fact sorted out specific divisions. We sorted division 0, 1, 3, 5, and cells that have gone through 9 or more divisions and acquired the CD138 marker. We were able to isolate 20,000 cells at each division. And our first step was to analyze these cells by qPCR to look at what the gene expression profile was of known markers of plasma cells and B cells. This is a heat map showing the percent 18S of biological triplicate for division 1, 0, 1, 3, 5, or 9, with red indicating higher expression and green indicating lower expression. One of the things we noticed was that there is a group of genes, and that there are distinct patterns to these gene expression changes, such that there are genes here which are induced as these cells differentiate to plasma cells, genes that are repressed as these cells differentiate, and genes that are turned on at the activation stage but are eventually repressed. And these could be divided out into 6 basic patterns of gene expression, such that you have progressive changes here, where genes such as EZH2 are continuously upregulated as these cells differentiate. And you also have genes here, such as CD28, which is a cell surface marker, that only comes on at the end in the differentiated state. The same can be true of both the repressed genes, where you have progressively repressed genes, and genes that are repressed specifically at the end of the differentiated cell type. And we were curious in understanding what the molecular epigenetic mechanisms were that were controlling these different patterns of gene expression during the divisions and differentiation. So epigenetic mechanisms that control gene expression primarily consist of histone modifications and DNA methylation, with different unique combinations of histone modifications, such as histone H3K27 acetylation, H3K4 monomethylation present at enhancers, with H3K27 acetylation and K4 trimethylation present at active promoters. These methylation marks are lost in the repressed state and replaced by methylation marks like K27 trimethyl, and the acetylation marks are also lost. And so unique combinations of histone modifications can tell you what the potential is at a locus and whether a gene is primarily on or off. Another surrogate for this, however, is the accessibility of chromatin to DNA binding proteins, whereas genes that are on and enhancers that are on are typically accessible, whereas enhancers that are off are typically losing accessibility. Now, this has primarily been measured by the accessibility of chromatin to DNase enzyme. However, recently, the development of the ATAC-seq assay has changed how we measure chromatin accessibility. If we wanted to measure different combinations of histones, we would perform ChIP-seq, which requires roughly 10 to the 6 to 10 to the 7 cells per antibody. Here we would need up to 4 antibodies to accurately measure the different chromatin states. However, a ATAC-seq assay, which reads out chromatin accessibility, can be performed from 1 to 50,000 cells, showing high levels of sensitivity and is in the cell range that we can isolate. This method was developed by the Greenleaf lab out of Stanford in 2013. And it works by taking advantage of the fact that a transposon loaded with sequencing adapters can only integrate its payload into accessible regions of DNA, such that treatment of cells with a transposon tags and primes accessible regions of DNA with sequencing adapters. DNA can then simply be purified, PCR amplified into a sequencing library, and sequenced to map accessible regions. This assay is sensitive and efficient, and it can be completed in about half of a working day. Provides a readout for the epigenetic state, such that accessibility traditionally means active, or inaccessibility means inactive, and you can identify enhancers, promoters, the DNA binding protein footprints, and nucleosome positioning, since these both protect the transposon from integrating its payload. We profiled the same divisions we did by RNA with the ATAC-seq assay. Here you're looking at the change in accessibility at 31,000 accessible regions between divisions 0 through 9, with red indicating high levels of accessibility and green indicating low levels of accessibility. We found that there were regions here that were accessible in the division 0 that lost accessibility as these cells moved to plasma cells, and regions here that were not accessible at division 0 but gained accessibility as these cells differentiated. We identified about 1,000 accessible sites that were specific to division 0, 1, 3, or 5, but around 6,000 sites that were uniquely accessible to division 9. A principal component analysis looking at how the variation between the different accessible sites and the cell types showed that in PCA1, division 9 was the most unique from divisions 0, 1, 3, or 5, but that there were unique differences between divisions 0, 1, 3, and 5. And this was surprising to us, and we did not previously understand how the chromatin accessibility landscape was remodeled very early and progressively, but that the largest amount of variation occurred upon differentiation. Since ChIP-seq is not in the range of the cell types we isolated, we were able to use ONCODE data and other publicly available data to annotate our accessible regions and provide functional, such that unique combinations of histones, such as the presence of H3K4 trimethylation with H3K27 acetylation, denotes expressed transcription start sites. We have poised enhancers, which are marked by only the presence of H3K4 monomethylation. We have a host of sites that had no known modifications and represent novel accessible sites to plasma cells. We have active enhancers, which are marked by H3K4 monomethylation and H3K27 acetylation. And interestingly, these bivalent regions, which have both the repressive and active histone marks. And it was these regions that we wanted to follow up with. So bivalent chromatin domains are typically found at regions that have both the dual presence of active and repressive modifications on the same nucleosome. And these are typically the active H3K4 mono or trimethylation in the same nucleosome as the H3K27 trimethylation marks. These allow for rapid induction of gene expression and often regulate developmentally important genes. The repressive modification in H3K27 trimethylation is deposited by the enzyme EZH2. In the recent 5 to 10 years, the histone and epigenetic modifying enzymes have seen a lot of target as drug targets for drug companies, such that there are about 50 to 60 drugs that are currently in different phases of testing in clinical trials. Most of them are targeted to the histone deacetylase inhibitors, but there are those that target the histone methyltransferases. GlaxoSmithKline has developed one, GSK343, that specifically targets EZH2, and we're able to use this and get our hands on this to look at what the effects are of H3K27 trimethylation in our system. And we simply wanted to ask the question, if we inhibit EZH2 using GSK343, what is the effect on B-cell differentiation, and does this have any impact on the timing of gene expression? We were able to take advantage of an ex vivo cell culture system here, where we take B cells out of a mouse and culture them in the presence of LPF and IL-2 and IL-5 cytokines for 3 days. And we know from previous history in our lab that we get a very robust differentiation. And so we added the GSK343 into the system to test what happens in the presence and absence of this inhibitor. We were able to show that with increasing amounts of GSK343, we specifically inhibit by Western the H3K27 trimethyl mark, but do not inhibit other methylation marks such as H3K4 trimethylation or total histone levels. When we assay the effect of this drug on differentiation by flow cytometry, we get about 9% of our cells differentiate to plasma cells after 3 days. However, in the presence of GSK343, we see about a 2.5-fold increase in the number of plasma cells formed after 3 days. This is quantitated here on the right. We performed RNA-seq on these cells to analyze what the gene expression changes were in the presence of GSK343. And if we look at the annotation of genes upregulated, we see that the protein secretion and the unfolded protein response are some of the processes that have been upregulated. And this is known to occur in plasma cells as they secrete such high levels of antibody it causes stress on the cells, and they upregulate the unfolded protein response in order to deal with that stress. We also find that they downregulate and change their metabolism, and this is also unexpected in that we did not know how these cells metabolically change from a quiescent to an active cell type. We also find that key plasma cell transcription factors are upregulated, while the key B cell transcription factors are downregulated. And chief among these factors was the BLIMP1, which is a master transcriptional regulator of plasma cell fate. The BLIMP1 was also interesting to us in that if we look at the BLIMP1 locus, and here you're showing the two annotated promoters for BLIMP1, and each of these tracks shows the signal for attack-seek at divisions 0 through 9. Here you can see a peak of accessibility at both BLIMP1 promoters, and both promoters are accessible at division 0 at a time when the BLIMP1 is not expressed. However, BLIMP1 is expressed at division 9, and so we wanted to understand what was regulating the timing of expression despite the promoter being accessible at earlier divisions. So we turn to our ex vivo system, and we turn to a mouse that contains a BLIMP1 YFP reporter developed by Sue Keck's lab. These mice have the BLIMP1 promoter with a YFP transgene introduced so that there is a fluorescent readout for expression that mirrors the in vivo BLIMP1 expression. We labeled these cells with the division dyes, CTV, so that we could also track the emergence of YFP expression in relation to cell division, and then treated them in the presence or absence of GSK343 with our differentiation cocktail. We found that division on the x-axis here versus BLIMP1 YFP expression on the y-axis, we said that these cells go through cell division, but that in the presence of GSK343, we have about a 2.5-fold increase in the amount of BLIMP1 YFP positive cells, and that if we look at the BLIMP1 YFP expression by division, that the GSK343 treated cells express BLIMP1 in an earlier division than the control cells, suggesting that EZH2 and H3K27 trimethylation regulate when BLIMP1 is expressed developmentally. So our model here is that normally during B-cell differentiation, after activation, these cells proliferate for a period while they undergo chromatin remodeling prior to terminal differentiation, and that EZH2 represses BLIMP1 such that these cells can proliferate and before terminally differentiating. In the presence of the inhibitor, EZH2 is repressed and these cells divide more but also terminally differentiate more, such that B-cell differentiation is coupled to the rearrangement of accessible chromatin, and EZH2 seems to regulate the magnitude and timing of plasma cell differentiation. And this may allow for processes such as selection and regulation of the magnitude of an antibody response to be regulated in vivo. And this is also interesting to us because epigenetic therapeutic agents can be immunomodulatory, and that treating the patients with these inhibitors is going to have effects on the immune system. With that, I'd like to thank the members of our lab who contributed to this work and our collaborators at both Kappa Biosystems and at the Inaki Sands' lab, the different course facilities, and our lab has positions available. And like that, I thank you very much for listening.
Thank you for those informative presentations. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, are all histones cytopathic and equally? And this would be for Carlos.
Hello. I show in my results that histone 3, at least for the bronchoepithelial cells, are more cytotoxic than the other histones, but there is evidence that histone 4 is also highly cytotoxic, and followed by histone 2b. And when the histones are complex as a nucleosome, they still are cytotoxic. Even if they are wrapped around with DNA, they are still cytotoxic. Can you go?
Oh, sure. The next one is for you, too. Were you done with that one?
Yes, I'm done with that one. You can go to the next question, please.
Next one is for you, too. In addition to H3.3, are other histones toxic and increased in plasma or extracellular compartments in patients?
In the case of the COPD, we also look for some other histones, and we were able to see it, but the major difference was in the histone 3.3, and we found this one was hyperacetylated, which is also resistant to be degraded. But in other disease, the whole nucleosome complex, which include the 4 histones, are also present in the bloodstream, and they are cytotoxic as a complex too.
How do the DNases inhibit the H3.3?
The example of DNase as an inhibitor is specific for the autoimmune disease, because when the cells release DNA, when the cells die, they release nucleosomes wrapped around with DNA, and the nucleosomes protect the DNA to be degraded. Then, by treating with DNase, you can avoid this connection, which is the combination of histone with DNA is like the source of antigens for autoantibodies. Then preventing the DNA to be there by treating with DNase, you will be reducing the possibility of expose new antigens. But the DNase will not prevent the toxicity of the histones.
How are the histones released out of the DNA or nucleus into the cell?
They are, as I showed, at least 3 different ways. In neutrophils, during the process of netosis, when they release the histones, they occur several enzymatic digestion of the nucleus and modifications of the histones. These histones become citrulinated, and then they eject the whole nucleus outside of the cells, and the cells disappear. That's one thing. When the source of histones are cells that are dying, like during apoptosis, the same thing happens through caspase or other enzymes that release, first they degrade the nucleus in a small part and vesicles, and then start releasing these small vesicles outside. And during necrosis, the whole cell break apart and release the cells, the contents of histones and nucleosomes.
Are all the hyperacetylated histones not DNA bound?
The acetylation group bound specifically to a lysine, which is a very basic amino acid residue, and a lot of lysines are in the tail of the histone tree. Then normally this lysine, when it's not modified, is interacting with DNA because of the charge, is positive charge, and is interacting with the negative charge of the DNA. But the modification that is induced by the acetyltransferase, when this acetyl group is attached to the lysine, they become positive, and then they repeal the interaction of the tail of the histone from the DNA interaction. Then you will assume that at least that tail is not anymore like hugging the DNA and holding the DNA, and is free of that interaction at that part. But the histone itself, the histone will still assemble into the nucleosome, and the DNA can be interacting in different places. But this makes more accessible the DNA to be used for the transcription machinery to work.
Does deacetylation of histones reduce inflammation?
There are several studies, and at least there is one drug that is claiming to enhance the activity of the histone deacetylated. and they are a mice model, and in patients they are using this drug that is increase a little bit the activity. In that way, the acetylations are reduced, and they show that they are improvement in the symptomatology of the patients. Then we can deduce that they will be a great significance if we are able to completely abolish the hyperacetylation of histones in COPD.
This next one I believe is for Chris. How different do the CHIP experiment results look depending on the quality of antibodies?
Yes, that's a very good question. The quality of antibody is probably the largest determinant of success for a CHIP reaction. Our lab, when we're developing CHIP assays with new targets, whether it's a histone or a transcription factor, we routinely buy 3 to 5 commercially available antibodies and test them all at the same assay and find very different results depending on the different antibodies. And so finding the highest quality antibodies in your assay is very important for success in CHIP experiments.
Chris, I believe this next one would be for you too. Is it known if overexpression of EZH2 changes the total modification of histones?
We don't know currently if overexpression changes the modification of histones. We know that EZH2 is upregulated in our system, but our theory, we believe that it's more recruitment of EZH2 to specific sites that changes the modification versus a global upregulation of the modification.
Okay, and Chris, this next one's for you too. Might your work on plasma cells provide potential therapies for multiple myeloma?
Yes, that is multiple myeloma. We also have projects looking at autoimmune diseases where plasma cells are a component. So this is very much a goal of ours, is to find biomarkers and potential therapeutic targets for these diseases.
Okay, the next one is for you, and it's a 3-parter, so let me know if you need me to repeat. What is the advantage of ATAC-seq over RNA-seq? What other information can you get since you already have 5 sets of genes? And why don't you go direct to histone modification analysis? Do you want to just take them one at a time?
Sure.
What is
So
the advantage of ATAC-seq over RNA-seq?
Sure. ATAC-seq and RNA-seq give you 2 very different pieces of information. RNA-seq tells you the expressed set of genes and what the level of expression is. And so that's an mRNA and an RNA assay. ATAC-seq is a DNA chromatin assay that tells you where the regulatory elements are in the genome and if they're accessible or active or not. And so RNA-seq will tell you if your gene is active, and ATAC-seq can pair nicely with that assay to tell you where the regulation of your gene is. And so the other information we can, so since we know we have 5 or 6 classes of genes by methods like RNA-seqing, real-time PCR, what we don't know is how those genes are regulated differently by classes, such that the regulation may be different for genes that are upregulated progressively versus a gene that I showed like LIMP1, which needs to be upregulated at a very specific timing during differentiation. And so we don't go directly to histone modifications, mainly because of cell number limitations. And here we're working with 20,000 cells per assay, and oftentimes we have to work with a few thousand. And ChIP-seq currently is not robustly ready for those cell numbers, whereas ATAC-seq and RNA-seq are. And so those are the assays we turn to.
Okay. The next one is, I am looking at a gene promoter region, DNA methylation. Healthy cells usually have very low methylation there. If my toxicant only decreases the methylation even further, is further decrease in methylation significantly measurable using sequencing? Let's see, who wants to take that one?
I would say certain sequencing applications for DNA methylation are dependent on the amount of methylation present. Enrichment-based assays may not be the best way here, but so bisulfite sequencing-based assays would not be sensitive to how much methylation you have and would provide you a digital readout for the amount of methylation you have, either at a specific locus or genome-wide.
Okay. Well, we have run out of time today. I would like to once again thank Dr. Kim, Dr. Barrero, and Dr. Scharer for their presentations. Do you have any final comments? Dr. Kim, Dr. Barrero, or Dr. Scharer?
No comment. Thank you so much for the time and for your
Thank you for your attention.
Thank you.
Okay, I would also like to thank our sponsor, Rockland, for making today's educational webcast possible. This presentation has been approved for continuing education credits, so if you want to obtain the credits, click on the Get Your Free CE Credits button located in the lower left of your screen. This will take you to a page listing all of our speakers and presentations. Please select the CE button under the presentation and follow the instructions to claim your certificate. Today's webcast will be available for on-demand viewing through April of 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.